Yes, so my name is Rajmund Mokso and I work at DTU Physics currently. And this work that I will present, many of this has been done, much of this has been done also but by my team here at the DTU, that is Emil Wisby is doing his PhD, and Francesca, who was the research assistant until recently. And so this was uh, also together with other people, which I will then acknowledge later on in the talk. So my idea for today was to tell a little bit about a very application-driven approach to do X-ray probing of, as I call, challenging samples, and you will see what I mean by that, and do it in a multimodal and multi-resolution manner. And what are there the challenges, and should we be afraid of doing this? Because, of course, we cannot be expert in every type of X-ray method. So should we, question is, should we just focus on what we know best, or should we try to really try to answer the questions which came up, which could be requiring us to go into fields which we are not feeling very comfortable. So this is the case here, what I will present. And I do this with the following outline. First, I'd like to introduce you to the some new approaches of sustainable delivery of nutrition to crops. This is the project that I will explain. And on that project, I will go into several of the methods. First, I'd like to talk about how we do this with using laboratory microtomography that we have here at DTU. Then do what cannot be done in the laboratory micro CT. We go to the synchrotron and do micro and nanotomography, and also then switch from time to time to do spectroscopy so that we are sensitive to the elemental distribution and also some chemistry. And then finally, lately we also had to do small angle scattering because the questions that we asked we were not able to answer them during using the techniques that I mentioned previously. So this also forms the outline of my talk, and I'll try to be quite clear when I'm talking about which method. So hopefully that will be fine. And first of all, so where I come from, so I work, as I told you, in Copenhagen in the DTU Physics, Technical University of Denmark, and more precisely, we are part of what we call 3D Imaging Center. It's a national competence center on X-ray and neutron imaging. And that competence center means that we have a know-how and about different X-ray techniques, and also we do development of this method, but we are also drivers of some of the scientific uh, projects that then use the X-ray techniques, different X-ray techniques. And these projects are, as you see here, in the terms of university, we call it university users because we are also a center where you can just come and make your beam time like in a synchrotron, but in a lab. And we do it for different types of materials and also biomaterials, and natural cultural heritage is currently a very big driver for actually building one of our new instruments here, which is completely dedicated to that. But I will not talk about that so much today. I will mostly focus on the bioimaging part today. And one of our missions is also education. That's actually a very important mission that we have here. We can in our facilities, and we do in our facilities, host several courses, X-ray science courses, that at any moment we can actually have a hands-on session for about 40 students at the same time, dividing them to 7 different instruments that we are running today. So this is also our strong point. And then so the Technical University of Denmark is on the other side of the Öresunden links is, which is the organizer of this webinar. We, however, have a strong link to Sweden, to the Max IV laboratory, where we both run a beamline, the Denmax beamline, more precisely the imaging part of that beamline, so the tomography part, where you will see some results from there. But also, we do care about the data analysis strategies of users in general from Max 4. And I also come back to this at the very end. So we have this strong link, and we have a number of instruments. I will not go into detail here. That's not the point. That was all what I wanted to just say as an introduction from where I come from. And now it's time to talk about plants. Plants is and this is one of the challenging systems that I said in the title. So why plants are so challenging? They seem to be not moving much, but still they are alive, and we want to investigate what happens in the plants alive. So that you will see brings us to many challenges, and in more particular, the problematic we are trying to address is the following. So, first of all, we have to rethink the way we are giving nutrition to plants today, and the reason is very clearly the growth of population, the degradation of the soil, because the fertilizers are inefficient that we use today. So that means that when we are giving nutrition to plants, we put this nutrition in the earth. The plants take up 10 to 20% of this, and the rest stays there or is washed away and goes into the water system. So that's not sustainable, and we want to figure out how to change that. I actually also this last week I have seen a projection, or how I would call it, where it is said that if every person on the earth would have an ambition to live like the same standard as we have here in Europe or in Sweden specifically, it was the case, then we would need four Earth planets to sustain, to give nutrition and all what is needed for this type of life. So, okay, there is several different messages one can take away from that, but my message from this is that we need to anyway look into more smart ways to do agriculture, and this is the projects that we started 2 years ago together with Copenhagen University and the McGill in Canada. That is a Novo Nordisk project where we have to look and figure out what happens in plants if we deliver them the nutrition in the form of nanoparticles directly, not through the root system, through the earth, but directly on the leaf. So that's the project. And to answer the questions, how, and also to look in forward how this all can be done in the future, realized, we first thought that we will be fine with doing just X-ray microtomography that was original in the project, but then we realized that it's not actually, that's not enough. So we realized we need to do both X-ray fluorescence imaging and also SAX imaging, and then try to combine all this together. Now that is the big picture, and now what we do in more particular, we have selected some model plants. This is barley, soybean, and tomato. And now actually we have a potato. We work a lot also on potato plants, and we are developing specifically DTU energy part of the project, developing nanoparticles, different nanoparticles, and we are looking into how these nanoparticles are able to go and be transferred in the plant and be delivered in the right place in the plants. Now that maybe you ask why nanoparticles. So the reason is that some of the nutritions that we would like to deliver to plants are not mobile into plants. And especially if you are applying them through the leaves, which the leaves are not made for taking up nutritions. They are made for providing air to the photosynthesis. So, and then we have to, in a way, uh, mask that the particles that we are bringing to the plant so that they become mobile and then they are transferred to the places where we want to have them. So to all parts of the plants from that part. So now that is the overall goal. And now, where are the questions, the scientific questions that are not understood today? So there has been observations that if we put on the leaf nanoparticles and solutions, nanoparticles in solution, then at some point people were observing that these nutritions they are to be found in other parts of the plant. But how are they transferred there? That is pretty much unknown. And I show here a picture uh, from the PI of the project, that is Søren Husted from Copenhagen University Plant Science Institute. He shows that there is two hypothetic pathways that the nanoparticles can go in through the stoma. So these are the openings or that where the plants breathe. Then there is a white is the air, and then the cells you see here, and that either these nanoparticles will travel through this pore network and reach the transport channels in the plants, the phloem, and or they will somehow be taken up by the cells and transferred inside the cells into this. So now, which one is which one is the pathway that actually happens in the nature now if we put these nanoparticles in? So that is our principal questions, and now we need to figure out how to address this question. So what we do is uh, first of all, expertise of our team here at DTU is imaging. Therefore, if so, imagine so we have a sample, and then one of the signal that we can get out of it if you shine X-rays on it is the direct absorption or phase contrast, and that will tell us 3D microstructure information. It's possible to do in vivo, and we can, for example, one of the examples that we can do very well is to look into fresh and hydrated samples. Now, the second, I will show you why, but it turned out that it's not sufficient. So the second thing we looked at is using X-ray fluorescence and near-edge spectroscopy that add additional information, and which is the chemical composition of the sample that we are looking at. So that was important. And finally, we also figured out that all this was not enough, and we also needed some additional information about statistically how these nanoparticles are distributed in the leaf, and that could the small angular scattering mapping give us. Now uh, I will shortly introduce all these techniques, each on one slide only. So that's not the purpose here, but only on one slide. And so first of all, state-of-the-art laboratory microcity that we are running here at the DTU Physics. And there we have a beam that is a cone beam geometry. So we have a sample, and putting a detector somewhere behind the sample gives us X-ray magnification. In addition to X-ray magnification, we also have a visible light magnification that is connected to the detector. And so here you can see the source, and then it goes, then the sample is placed here. In our case, the sample is placed on a in a number. We have developed a number of specific, and this is I will go into more detail later on. But we have developed a number of specific sample environments. This is the first generation here. What you see, where the plant is in the scanner. It's alive. The root is placed under here, and then it's during the scanning, it is hold still in place so that we don't have what the motion artifact that we would have if the plant would be just floating, the leaf would be floating around. So with this technique, we typically working with 2 to 10 micron spatial resolution, scan times in a range of an hour, and field of views 1 to 20 millimeters, quite a big range of field of views we could do because we also have different scanners. So that's the reason for this large range of field of view. And we do use phase contrast or in the lab, also moderate, but we do use it. And what is very good in this is that we actually have enough time to do modification if we realize that something should be better optimized specifically in terms of sample environment. So this is an important point doing experiments in the lab. Then we used synchrotron tomographic microscopy, where we have a parallel beam geometry. There is no more X-ray magnification, so all magnification happens with the visible light microscopes. The sample is sitting in front of the microscope as usual. Typically, we are talking there about a micrometer spatial resolution. Scan times, now you see here, it's more in a seconds than in an hours. And the field of view 1 to 2 millimeters if we do it as in our case in Max 4. And then we have a very nice phase contrast, of course. And however, there is one thing that is really very challenging in terms of making any modifications if we realize that something does not fit or doesn't work. So that means one needs to be really prepared well for in this case. Then quickly about synchrotron nano tomography, nano holotomography we call it, or the nanotomography in general. Can we do it in a synchrotron? We can do it in two ways. Going back to this cone beam geometry again by focusing down the parallel X-ray beam to a small spot. And this way we could achieve again X-ray magnification in addition to the optical magnification and have a very high spatial resolution well below 200 nanometer, several minutes scan time, and a field of view of few tens, hundreds of micrometer at the best. And this gives us excellent phase contrast, much larger contrast than in the parallel beam geometry. And however, our biggest enemy is the radiation damage in this case. So and the last two is quickly X-ray fluorescence. In this case, we focus down the synchrotron beam again. Not doesn't have to be the very fine focus. It depends on the specific sample needs and so. And then, so this image actually doesn't show it very correctly because I realized that I switched the wrong image here, but we have a fluorescence detector here that will detect the outcoming, the emitted signal. And this way we can map some relatively large pieces of sample and sense what elements are contained in the sample, but also in what chemical state they are. So this is quite important additional information to the tomography. And then finally, small angle scattering. And here we again focus the beam down to our sample. And then as you see, there is a long flight tube at the end or somewhere in the flight tube, there is a detector, a large detector that will detect the scattering signal. Here we can also quite quickly scan relatively a few millimeters of the leaf size, leaf structures. And then typically it's even faster, but if we do a big field of view, then it takes a few minutes, maybe an hour or so. And also the spatial resolutions are a few tens of micrometers. And this gives us statistical information about nanopart at the very scale of the nanoparticles, which is a few tenths of nanometers in this case. So summarize this first part. We have we can use different geometries of the X-rays, and we can access the micro scale and nano scale. We can do it in a multimodal manner. Specifically, if we focus down the beam and map the samples, then we can record both the fluorescence and the spectroscopic signal, and the scattering signal. And so that is provided. So these are all different methods that are used in number of beamlines, and I will tell you which beamlines we are using specifically. So first of all, big part of this presentation will be about tomography. So just to uh, maybe remind or maybe have a quick introduction to this. So when we have the X-rays coming on the sample, then we do rotate the sample, and while rotating, we acquire tomographic scans, radiographic projections, and that's what consists. That's the tomographic scan consists of these radiographic projections, and then based on those, we can do the 3D reconstruction. We can do even movies of samples that are moving, or look at slices and in any position that we are interested in. And so this is the technique that we are using, and we can use it to look at various scales from micro tomography, micro scale. So here you have the whole leaf, barley leaf, and we can zoom into a specific region, do with nanotomography, uh, in, and look into the cells, and we can zoom in further and look into just, for example, the chloroform, and then look into these subparticles structures. Uh, I will talk about it a little bit later. So what we have to be very um, aware of, that with the improving of the spatial resolution, there is, as I already mentioned, radiation dose. It's also increasing. So we go from single grays in the microtomo to thousands and even millions of grays in the nanotomo scale, in the nano scale, and then also the sample sizes become smaller, and the 3D scan time is also getting longer. So these are all this means that for specific questions we need to really think about all these four uh, parameters. Specifically, if we are interested into imaging material that is sensitive either on time or on radiation or both of it. So what we do for this project with our laboratories is systems, and how we do it. So then we use mainly our two micro CT systems, commercial ones, and we are also starting soon to use our home build nano CT that is coming online in December, and is specifically so we are often very application driven here in the 3D imaging center. So we are building actually this scanner specifically for this project, but it could be used for other things also later on. So for to be able to scan these leaves alive in our scanners, there has been a lot of study on how they behave, and what we can allow ourselves in terms of time and the radiation dose, and what type of resolution spatial resolution we can then achieve in that. So the main point is to keep these plants happy. And happy is difficult to know if you are talking with, if you are not able to talk to that sample, which you are not because it's plant. So it's difficult to know in the beginning what means that the plant is happy, especially for us physicists. But we little by little figured out. So we know that what is important is light. We also know what is important is humidity. Therefore, we make chambers where the plants are inside, and there provide constant humidity. But we also know that for the scanning, we also need to immobilize them. So so it's a trade-off of all possible parameters that we try to combine here, but it works, and we can achieve. You see here scans with even sub-micrometer pixel size. The resolution is still about 2-3 micrometers, but in about 90 minutes. So you see here the cross sections through a leaf and shows very nice details of the cells. But we also put a lot of effort into studying. You see the plot here of X-ray exposure time and the thickness ratio. Thickness ratio means that when we start to see collapsing of cells from the initial state to and here we do a number of scans, so we increase the time, exposure time, and we follow how these cells collapse. And then this tells us a clear information. So if we do it with for many different samples, it tells us clear information to what point we can follow until, for example, can we do 1, 2, or 3 scans of the same plant in vivo? Because this is what we need. We need to follow how these nanoparticles are taken up. So we need to do multiple scans, not only one. So this was goes first, and it's very important also for the synchrotron studies that you already have a feeling how these systems behaves when you are scanning. But it's not only the dose, but it's also the time that the sample is in the scanner. And here is a visual representation of the same thing, where we have tomography reconstruction of the sample in the initial condition, and then after a few hours, 4 or 5 hours of scanning, you see this all the leaf is collapsing. One of the ways we can go around this is that we actually use as much as we can the phase contrast properties of the modern scanners that currently we can do. And that phase contrast reconstruction gives us both, for the same time of exposure, gives us a much better image quality, or it gives us the same image quality for shorter exposure time. So this is actually very important. Without this, we would not be able to scan live plants. We know we are not able to scan live plants in our laboratory systems because of the time, but also because of the dose. So I will skip this. Actually, this is just to demonstrate the phase contrast. And now I will show you some of the results that if we manage to keep these plants stable with the different sample environments that we have, then we can observe clusters of nanoparticles. Of course, not individual nanoparticles, which are tens of nanometer large, but only clusters. So where do they cluster? Which places of the plants? And here you see one visualization. These are two leaves scanned at the same time. One is a control, and one is with applied nanoparticles. And then we can see some clustering, and that's the purpose, to see where these clusters are in the leaf and follow them. Uh, Here, this movie shows something that we need to understand actually before we look in the nanoparticles, and that's the water film, that the nanoparticles are suspended in this water film. And this water film has to be on, that when we apply to the surface, it has to penetrate inside. So you see with the arrow, you see where the water flows inside. Uh, and it can only flow inside if this stoma, these breathing channels are open, and then they allow this water to flow in. And that is a further big challenge in the case of this scanning of plants, to be able to control that the plant is happy, and the plant is happy, then this stoma is open. I mean, this is a very rough statement, but it's in some senses it's true. So if these stomas are open here, then one can observe that the nanoparticle solution goes inside. So that's the first point. And then the next step will be then to observe what happens after, but we don't have a resolution enough with the lab to observe what happens after. So we can do two things. we can build our own nano imaging system. As I told you, this we will have ready for in December. We can also do another thing. We can go to the synchrotron. So first we went to, we did some imaging at Denmark for a specific here. So before I go there, so of course at the synchrotron, we even, as I told you, we even have a higher phase contrast than on lab. So phase contrast, we achieve it by just moving the detector farther away from the sample. So this is something one needs to optimize when you come to the beamline. And then that gives us also much higher sensitivity to the structures in the leaf. We, for that, for the beam time, for example, we do prepare on-site these, we have this specific sample holder in that specific geometry of the leaf, where what we do is we actually apply a drop directly here on the surface. And this, in addition, it's closed, the chamber, and there is a CO2 inlet and outlet, which regulates how much CO2 the plants have, and that regulates how much the stoma is open. So that is also quite sophisticated. And then we also we can put that on the rotation stage and do tomographic scanning. And what we could observe in this case, that when we apply these drops, in this case there was a slight iodine enhancement of the contrast, because we wanted to make sure that we can clearly see the water contrast to the cells. Then this we can see how this penetrates and goes in. And that means that we succeeded to actually keep this stoma open. And that was thanks to all these studies that we did in our laboratory before we went to the beam time. So that was extremely important. And then we still, of course, here we can see how this solution goes in. We still cannot see the nanoparticles and even small clusters of them. So therefore, we go and do nanotomography at the synchrotron. And this we did at ID16B mainly, and some work also at Spring 8, but that was very preliminary work. So the live imaging of the plants at the nanoscale was ID16B. And here again, I remind you that the idea is to follow how these nanoparticles, when they enter, so we know now they enter, but we want to follow them and then what happens later on. So here you see a tomographic slice from the with 50 nanometer voxel size of a fresh live leaf. And you see some water film. Here is the stoma, and that stoma is closed, so there is no film in. And now I can tell you the story of this beam time, that actually so this is the team, actually half of the team that was awake at that moment when we did the photo. So, and then in this beam time, we have not managed to open the stoma for these plants that we have scanned. So we could not see um, in case of the barley, we could not see these nanoparticles going in the plant. Also, we made quite a breakthrough in understanding that we can image these plants with 50 nanometer voxel size live. So that is also something completely new. And um, so Emil is a PhD student who is just writing up this publication on that. We used here nano holotomography, where we focus down the beam, and then depending on where you put the sample out of the focus and the detector, you get a specific ratio of magnification. Here we have, you see, this is an example of some other cells where you can have a very high, we have a very, very high contrast. Actually, it's a holographic regime, so you need to employ specific tools for doing what we call phase retrieval to get the density map of your sample. But this one can do. And when it's on the sample, when the sample is on the in the beamline, it looks like this. And here you see this, as I showed you in previously, this plant holder. And here we had to be innovative because we don't have much space in this nano imaging setup. So the plant is upside down. So the roots are up here for that few tens of minutes when it's sitting inside this beamline, and then we do imaging of the leaf directly. So the X-rays go through here, and then the detector is here. So that was not showing us the nanoparticle pathways at this first session. We had one beam time for that because we could not at that moment still control the stoma. That since then we can, but what we could do in certain plants which were the nanoparticles were put on before putting it on the beamline. There we could control the stoma when the plants were still outside the beamline. There we did apply manganese oxide nanoparticles, and then we did a tomographic scan, and we didn't see them. And then we wondered, are they there or are they just completely dispersed as they should be, actually? So for that, to prove that, we came up with the idea to put calcium chloride that will agglomerate these nanoparticles. So this is what happened. And when we did this, we suddenly started to see all these agglomerates, clusters of nanoparticles. And in 3D, also one can visualize it. And that was then clear proof that the nanoparticles are in and they do as they should. They do not cluster because they should be dispersed. That's how they should travel through the cells. And at some point they would cluster, but this we couldn't reach there because uh, during this one beam time, and that's also the takeaway message, some goals cannot always be achieved, and the goal was to actually show that we can image these structures alive at that resolution, and then the secondary goal, and maybe for the next beam time, is to be able to follow these pathways as they are. We can also image at nanoscale much easier samples that are freeze-dried cells, plant samples, and then you can really go to the resolutions where sub 100 nanometer resolutions, and you can really look inside the chloroplast, for example. But then the question is, it's very difficult actually to answer our questions, which are the pathways of nanoparticles, but it can answer other type of questions about the plants. So that's we didn't follow this path anymore, but it's something that we could think about also in the future. So as I showed you, we didn't see these manganese oxide nanoparticles when they were not helped to agglomerate. So we were thinking what to do. They are too small, even for this nanotomography. So we went to 4 max and at Mach 4, and we did small angle scattering. This is the image of on the detector that when you put in a sample, you obtain some scattering, and then it's a completely different story than from direct imaging. You obtain curves which shows how much different parts of the sample scatter, and here you see a map, a 2D map of the total scattering. And here we selected in several regions where we think here are nanoparticles, and there we think there are no nanoparticles. And then here you see then scattering patterns. So based on that, one needs to then fit the model, and we were quite new in this field, and fortunately, my new colleague postdoc Cheng Pong took over this part, and then we learned a lot with the scattering when looking into, in this case, we looked both in fresh and live plants, but also in freeze-dried. I show you here for the result of a freeze-dried, which were dried in different time steps after applications, immediately, 1 day after and 2 days after. So it's not the same plant, of course, unfortunately, but we see the scattering of manganese oxide nanoparticles, how they change from 0 day to 2 days. And so we could actually see where they travel, these nanoparticles. And now the next step will be to do nanoimaging on the same samples. So, and the last thing I want to show you today in 2 minutes is just that we also needed to understand when we use the silicon carriers, silicon mesoporous silica shells as carriers of zinc oxide nanoparticles, because they could be very mobile in the plants. So we were able to show how these silica shells dissolve and how they release the zinc oxide. But this we can't do with the direct imaging and maybe with scattering at some point. But we first actually looked into the most straightforward method, and that's X-ray fluorescence and near edge absorption spectroscopy. So we did it at the Lucia beamline and in Soleil in Paris. And so we dosed the leaves, then we prepared from each part of the leaf, we crushed the tissue and prepared these pellets. So this represents parts, for example, the upper leaf or the lower leaf and so on. And then we obtained the curves of, as you see here, each point in this image has a spectrum, and that spectrum tells us about the chemical composition and also the chemical state of the silica. So when the silica nanoshells are disassembling or dissolving, then we see a change in the chemistry. And that could explain us different parts of the leaf. Here are we both scanned these pellets that I showed you, but also freeze-dried. These are freeze-dried leaf sections. And then in selected points, we did this chemical analysis, and that also explained us when and how these silica nanoshells dissolve in the plants. So that was all this multimodal information flow that we used in our project. One thing at the end I'd like to just highlight is that for both the imaging but also the other parts, it's extremely important to before one goes to the beamline, it's important to understand what one wants to do with the data, and being able to beforehand actually have maybe an example pilot data set where one can test out the workflow of the image analysis. That is very important. It's also very important to write this in the proposal for the beam time, because it is something that the reviewers look into quite a lot to, if you actually one knows what to do with the data and has the know-how. As I mentioned at the beginning, this is my last slide. In the DTU, we have also this what we call the Center for Image Quantification, and that if you don't know what to do with 3D imaging, you can ask for advice. Also, we have similar organizations also in Lund. So the KIM is also a collaboration between Lund, Copenhagen University, and DTU. So all this is so you can use different channels for that. And it's also, if you look at the web page, you can see the tools that has been developed, and you can just click on them and download and use them. And if you need more advice, then just ask us. And then the conclusions is that I think even though we don't have the experience in all, it's best not to be afraid of trying to do this multimodal, multiscale X-ray probing, because that can give us the complete picture sometimes. Maybe not the complete, but quite more complete picture at least than just deciding for one method just because we can do it the best way. And I think it's important that for this challenging sample, like that are alive, for example, that the key is definitely to spend enough time and resources to develop to the environments and the protocols that we can often easily do in the lab without going to the synchrotron. And then think about the analysis, what you do after with the data. And that was it for me. Thank you very much. And I'm looking forward for questions from your side.
Thanks. Thanks a lot for this great talk, Raymond. So now the talk is open for questions. So if you have questions, please raise your hands or or put in the chat. Probably there are already some questions.
Nothing yet.
Not yet. Okay. So if you have a question, please do it.
Maybe in the meantime, I can ask a question, Raymond. Very, very interesting, very impressive. I'm wondering a little bit if you can mention about, the team you're part of, and the background the team has, in, let's say, X-ray imaging. I presume there might be persons with knowledge in plant science, and also related to like the ideas you have to building these sample environments, really dedicated and keeping the plants alive and so on. Yeah.
Yes. Thank you for this question. I failed to introduce the team. In fact, it's very good. So yes, so this is the team, and so this is almost everybody in the team, not complete, but almost everybody. And more than half are plant scientists from Copenhagen University. Then maybe one, and then one fourth is a chemist or that this type of expertise where they develop nanoparticles, but also look into the influence of chemicals on the plant and on the soil and so on. And then the remaining is us doing imaging. So in our team, in the imaging team and DTU team, this is where we usually develop the sample environments. And while we have a very, very tight discussion always with all the group, and we meet always when we need to develop some sample environment. We always meet and we always make many iterations before we actually use the sample environment, for example. So the expertise we developed in-house for building sample environments for plants. But what is also nice is that all the people now in the team actually are able to do their own synchrotron experiment. And like this September, for example, there was one of the Denmark's beam times. We, from the DTU side, we were only helping out a little bit. So all has been done, and even in a specific sample environment for a modification of what we already had has been done by the people from Copenhagen University for their specific questions. So this team, as it is, has actually became all knowledgeable how to do synchrotron experiments in the process, during this process of working together in 2 years.
Yeah, super. Thank you for the answer, Raymond. We have a question in the chat.
Yeah.
What is the basic purpose to pre-train the samples before beamline experiments? So I guess with pre-train we can say pre-scanning, I presume, or so on.
Yes.
What is the basic purpose of that before beamline experiments?
Yes. So we actually do not do that at all. What I wanted to, maybe I was also not completely clear. So we never do pre-scans. What we do the development of the whole strategy of so we are not doing like we take a sample and we pre-scan it and then bring it to the synchrotron and scan it again. We never do this. So what we do, we really develop the whole concept of how we will do. If we cannot, first of all, we need to learn if we can get the information we want in the lab. That's the first thing we need to understand. If we can, then that's it. If we cannot, then we have to understand what we need to do to make possible at a given synchrotron beamline, for example, to do this. And that's what we do. So we develop the concept, we don't do pre-scanning. And that's, I think, what one should do. Of course, there are good reasons to do pre-scanning also. Some samples are, in our case, we don't have samples for that would need it actually. But in some cases, definitely there is a good reason. For example, today the modern synchrotrons like Max 4, they give you 1-2 millimeter field of view. So you like to have a larger sample, you want to know where to scan, then you do a pre-scan in the lab, and then you understand where you want to scan at the synchrotron with this 1 millimeter field of view. So that's a very good reason to do pre-scanning. We do not have that thing, but so I think, yeah, our strategy is to develop the strategy at the lab.
Thank you. And I can say the questions are not popping in the chat. So next question is how to ensure that the data analysis tool is functioning well before the beam time. So here I'm not sure if we're thinking, it's
Yes.
quite an open, question, image analysis or reconstruction. So feel free to answer.
Yes, this is an excellent question again. So there is honestly, it's very rare that we make sure that these workflows are running correctly. So, and it really needs a team in a way that works with these methods for some time. And I know that for example, also in plant science, there are teams that are able to that just go take the data from the experiment and they already have the scripts ready because they have done similar work before. We did similar things, for example. Maybe the key point here is to take something, to start with something that maybe has been developed by you or by others for maybe not exactly the same thing that you doing now, but it's similar enough. Like we do, for example, in the case of I also worked a lot with the foams. We developed routines to analyze cellular materials like foams, and then we did a campaign of imaging of different systems like food, for example. It was bread, for example, and there we could use with small modifications the already existing workflows. So I think this is the key, maybe to take something that works on a similar system that what your system does. And of course, by time, the workflows will be there in the team. And then every next experiment, you can already rely on those. But in the beginning, it's difficult. I'm quite sure it is almost impossible to find the time to make these workflows ready if you are new for that type of system before the experiment.
Thank you, Raymond. Then the currently last question in the chat, we'll see if there will be more ones. How do you distinguish leaf damage from perhaps motion artifacts? And then parentheses, if the sample is not entirely burnt and easy to see.
Yes. Yes. There, I think here I have only to say that we have no number that I can tell about this, but what we have, it's experience. So we know how motion artifacts looks like if the leaf would move, let's say into, like it would oscillate or it would move one direction. But what we can see from the images that it's a collapse. That's why I showed this plot. It's actually what we can measure is how, in principle, one of the parameters is how thick is the leaf itself after each scan. And if you see changes in the leaf thickness, then we know that the cells are collapsing, for example. And they could be because drying out if they are not correctly in the correct environment. But so that's so it's typically more like a shrinkage. If you go to nano imaging, that's the most typical sign of radiation damage is a shrinkage. That's always the first sign of radiation damage. So, but yes, but it's not always easy to distinguish from motion artifact. But I think I rely mostly on the experience, in fact, to be honest.
Yeah. Thank you, Raymond. I can let you know that the questions are popping in. We have two more ones. So question comes, we do not have any modalities available in our lab resembling a synchrotron. I guess we can say lab-based ones. We are new in the field. Can you give recommendation as to what we could do to near an appropriate approach before the actual beam time?
So yes, if you sorry,
go ahead.
Yeah, if you mean that let's say if you mean that you don't have lab scanners, this is what, if I understand correctly.
Yeah. Maybe for those of you, maybe Stina Hasselholt, if you would like to speak up, maybe you can take the question.
Yes,
directly, Raymond.
That would be great, yes.
Yeah.
Maybe.
Hear me now?
Yes.
We hear you.
Okay.
Go ahead, Stina, if you would like to develop a little bit more. Is it so that you don't have any lab scanners in your lab, I presume, right? Okay, we don't hear you that well, unfortunately. Okay, maybe let's anticipate that is the case, Raymond. And
Yes.
let's maybe recommendations to which lab she can go to, or
Exactly.
so on.
Yeah, that's so we have a. I don't know from where you are, Tina. Maybe you can also write it in the chat. But in a way, it is today. You find almost every university. You find a scanner or two, and then I think there are. In addition to this, there are some what we call, maybe you would call, core facility, if it would be typically this is how they call it for microscopic centers. And those we have also several in several places, not so many as individual source lab scanners, but yes. So it depends where you are. I mean, there is a network of these scanners that are available for anybody who would like to use them, and there is people have different systems how to access them, but it's possible.
Mhm. Do you want to say a few words, Stina? Let's see if your sound is better. Otherwise, we will move on shortly. Okay, then we move on to the next question. In your introduction slides, you mentioned that one of the main motivations for studying the nanoparticle absorption into plants is to fight soils degradation in agriculture. Nanoparticles are known to lead to cancer and in general toxic for human health. How is injecting nanoparticles into food chain an idea worth studying?
Yeah, that's exactly one of the questions that are to be answered in this project, and this is also why one team, 3 people, are working just on this. Not maybe completely on this in the sense that they are working more on how if these nanoparticles end up in the soil, what happens then? Because if if this is done properly, then all these these are nutritions, these nanoparticles. So this is simple nutrition, and that is nutrition for the plant, and that means that the plant will take it up, and there will be no nanoparticles after 3-4 days. So that is, let's say, in this sense, for the consumption, it would only be an issue if one would take it in a wrong time steps, or wrong times. Even like that, these are again nutritions. So we are talking about zinc that we take up anyway. So there is no because these are nanoparticles, it doesn't mean really anything for the health as it is of the people.
Thank you, Raymond. Next question. It's also in a way a general question. Let's see if you answer it, or if me and Lucia are in the room here will answer it. But is it possible for Beamlines to have some workshop to teach users even before beam time with some example data to learn about the workflow? I guess it's not a question to Raymond, but also to Links or Max 4. But please go ahead, Raymond, if you'd like to.
Yeah, I can start quickly. I think that's an excellent suggestion, and I know that there are, for example, I know that just an example at APS in Chicago, they are making a number of workshops, the beamline specific workshops, where they actually do teach how to deal with the data, but also how to prepare for the experiment. So I think this should be done probably. Yes, by somebody. Maybe Links is exactly the partner for that.
Yeah, we also tried to do this at the kickoff meeting of the Heritage Science team. There was after the, let's say, the seminar, the first day, there was a day devoted to beamline tutorials and so on. with the participant to the workshop, where we split people in different groups to show them some examples with spectroscopy and with imaging, showing them real examples of previously done experiments. And this was quite useful, and we had very good feedback, and we can go on in this direction, I think.
I think we just had that event again last week with students bringing in artifacts to scan and folder.
Yeah.
Right?
Yeah. I presume here we are not, when Tuna here speaks about workflow, we're actually speaking about like the scanning part. We're not speaking about data. Yeah.
Yes.
Exactly. Yes, there have been some, as you said, how to say, tutorials on spot at Max 4. What do we call them? Hands-on training, perhaps, at various Max 4 beamlines.
Yeah.
Yes.
We could do also some in laboratory systems. In case people are interested, we can launch preparing beam time also with practicals, not only with theoretical talks that are very useful, but also combining at the end with some tutorials. And
and then to let you know here, meantime, Stina has been writing in the chat and Raymond has been answering. So Stina was wondering a little bit about optimizing, the sample preparation, sample holders parameters, and mentioning that with a single beam time, how do you do this? You want to get the most out of it. And then Raymond, maybe you want to take your answer.
Yeah, so it's clearly impossible actually to do this during the beam. That's why I also mentioned it. So what I say that it's this is of course not an easy thing, but as I say, today we have a lot of possibilities compared to 10 years ago. We have a lot of possibilities to do very similar acquisition in the lab, at least what I know, the imaging wise, then we can do the synchrotron. And these labs are accessible. Our labs here are national facilities, so not only Danish people, but everybody can access it. So it's a question of just reaching out to some, and maybe that's the way, maybe. Or you, of course, you can ask. Yeah. So what we do, we know also another thing is that when we do this in vivo, this you maybe have learned today also what I mentioned. We do this, for example, the nano in vivo imaging. We didn't succeed with even the half of the objectives we had, but we understood, and then we still hoping to get a second beam time, clearly. So yes, so in vivo systems, I have never in my life managed to make anything work on the first beam time, even though I was preparing for it for a long time. So that's how it is, simple. And I think it's still possible to get 2 beam times, but you are not possible. Very probably you will not get 3.
Yeah. As a follow-up to that info, Raymond, I'm wondering if you could give some example of timelines of various projects you did, like how many months, let's say, or years did you spend preparing for beam time before you actually went, in the lab-based environment, before you went to the synchrotron, let's say?
Yeah, so when we started this project, we have not even thought about doing anything on a synchrotron. So our objective was to develop whatever we have to answer the questions in the lab. And so we developed all the methodologies, all the concepts in the lab. And then when we started to see the results, then we started to know clearly where are the next frontiers which we will not be able to reach with lab. And so that was 1.5 years after. So for 1.5 years we were just developing the whatever we could in the lab, but not really thinking about the specific beam time, but about the system itself. So we were addressing the questions and the system, and then it comes up very clearly, and then it's easy to also write a proposal because it's very clear then how you actually should what you want to really get out from the beam time.
Yeah. And what would you say about time frame? Like prepare for me. Are we speaking about months or years or?
If you want to develop a concept how you measure your system, and it's a challenging system, for example, then I would say that usually if you do it in an intensive way, then if you in a lab, for example, from my experience, typically when you work 4 or 5 days, 2 times on your system, then you understand everything what you need to do for the time.
Okay. There are no more questions in the chat right now.